Biological data in clinical trials is messy and highly variable. To prove a drug works, researchers must force this chaos into a rigid, mathematical grid. This translation process is clinical data management, or CDM. Its objective is to ensure that every data point collected is accurate, complete, consistent, and strictly compliant with regulatory standards. Clinical trials face a persistent challenge. Real-world data arrives from dozens of disparate sources and is highly susceptible to human error. However, the statistical models used for approval require pristine, standardized datasets to function. Pharma companies bridge this gap using a 15-step data engineering pipeline. It's segmented into 3 zones: architecture and setup, the cleaning engine, and finalization. This architecture dictates how raw human inputs are systematically filtered and refined into locked, regulatory-grade outputs. Statistical truth in clinical research cannot exist without structural data integrity. To understand how drugs are proven safe, we must map out how this blueprint engineers that integrity from the ground up. The process begins with Phase 1, study startup. This is where the physical pipeline is constructed before the first patient is enrolled. The foundation is the Data Management Plan, or DMP. This master blueprint details exactly how every single data point will be collected, handled, and verified throughout the study. Designers use that plan to build case report forms with logic gates called edit checks, programmed to instantly reject impossible data. Before go live, user acceptance testing runs dummy data through the system to confirm these gates function exactly as specified. Once the database is validated, the study enters steps 2 and 3, data collection and data entry. These are the primary intake valves for the system. Inputs flowing into these valves include physical lab results, electronic patient-reported outcomes, electrocardiograms, and telemetry from wearable devices. Site coordinators enter some of this information manually into the Electronic Data Capture System, or EDC. Other streams are ingested automatically from external vendor APIs, removing the risk of manual transcription errors. This stage is orchestrated by the clinical data manager and physically built by the database programmer. Rigorous upfront database architecture is the only way to prevent contaminated data from causing a catastrophic failure during final analysis. As patient data begins flowing into the system, the pipeline shifts into Phase 2, the cleaning engine, where information faces continuous scrutiny. In Step 4, data validation, pre-programmed edit checks scan incoming notes. If the system detects invalid lab values or an age mismatch, it triggers a red alert. Manual and medical reviewers catch what automation misses, diverting logical inconsistencies into a quarantined holding loop. To resolve these discrepancies, the system relies on Step 5, query management. This is the formal, documented mechanism for correcting questionable data. A query follows a strict lifecycle. The data manager raises a query, the site investigator responds with a correction, and the manager verifies the fix. Once resolved, this corrected data is injected back into the main clean stream. Query management acts as the engine that forces messy real-world observations to conform to the rigid logic of the study protocol. Integrating external third-party data streams requires the pipeline to merge disparate sources into the central EDC without compromising the existing structure.
External data reconciliation ensures that files from central laboratories, ECG readers, and biomarker vendors map correctly to the patient IDs and visit dates recorded at the clinical site. The most critical integration is Step 8, SAE reconciliation, which tracks serious adverse events like hospitalizations. Serious adverse events recorded in the EDC must be perfectly reconciled against a separate pharmacovigilance safety database. Every detail, from onset dates to dosages, must align exactly across both systems to satisfy regulatory inspectors. Step 7, medical coding, standardizes human language. Different doctors use different terminology for the same symptom. These subjective descriptions pass through dictionaries like MEDRA or the WHO Drug Dictionary, emerging as standardized alphanumeric code blocks. A dedicated medical coder takes responsibility for this translation, ensuring the data is globally uniform. Data managers concurrently execute Step 9, data cleaning, and Step 10, interim data review, checking for duplicates and monitoring safety signals while resolving the final open queries. Multi-system reconciliation and coding elevate isolated observations into an integrated, globally verifiable data ecosystem. The pipeline now reaches Phase 3, finalization. This is the gateway that separates the data collection process from the final statistical analysis. Step 11 is the database freeze. This temporary lockdown halts all changes, so the clinical team can perform a final quality review on a static dataset. Once the freeze is successful, we reach Step 12, database lock. The system cannot lock until all queries are closed, coding is finalized, and vendor data is reconciled. After sponsor approval, the padlock snaps shut, severing all inbound data pathways. After a database lock, the data is unalterable. No further changes can be made unless the study follows a highly scrutinized formal unlocking procedure. With the data sealed, the pipeline moves to Step 13, data export. The database programmer extracts the finalized information into standardized industry formats, such as SAS, SDTM, and 8M datasets for the biostatisticians. The database lock is the defining threshold of the trial. It transforms the ongoing collection of subjective data into an objective, unalterable record. The exported datasets now travel to their final destination, where they provide the proof required for regulatory approval. Step 14 is statistical analysis. The biostatistics team applies complex mathematical models to the finalized results to measure efficacy and safety. They generate tables, listings, and figures, or TLFs, which provide the numerical evidence that the trial met its clinical endpoints. The 15th and final step is the clinical study report. This is the comprehensive written compilation of the trial's methodology and outcomes. This report forms the core of new drug applications submitted to agencies like the FDA. Without this document, the trial data cannot be used for regulatory submissions or medical publications. The full 15-step workflow shows how these roles interact. Database programmers build the frame, clinical data managers run the central validation loops, and biostatisticians extract the final insights. While platforms like Metadata Rave or Viva Vault provide the tools, they are empty shells without this rigorous operational blueprint to guide the data through its lifecycle. Every drug currently on the market reached the patient by successfully navigating this 15-step workflow, proving that medical breakthroughs are only as reliable as the data structures that support them.